be co-host tomorrow?
Yeah. Yes, of course. Everybody can be co-host. And no, Frank, you can't kick people out. I'm kidding, of course. You're eating a burrito, Frank? I haven't eaten any lunch. It's not fair. Lunch is at 12.
No, it's a hot pocket. It's a hot pocket.
Oh, that's even worse. I love Hot Pockets. I love Hot Pockets.
Philly cheesesteak.
Oh, I hate
That's
it.
decent. That's decent.
Yep.
Yes, sir, Jamar. Yes, sir.
You're gonna eat it right in front of me too, Frank. That's not right. Not right. Could someone, Frank, make sure you let those guys in.
Oh.
Oh, where is this thing? I just did it.
Jamar, what you eating on? Oh yeah, yesterday y'all was dogging on me, but when Jamar was eating, nobody said anything.
I had to call him out for you, Layla.
Nobody said anything. And Taylor was eating her noodles. Nobody said anything about that.
I said something. I said something.
Only Mr. Mendoza said something, but when it was me, everybody was in the chat and they was on me. Ain't nobody stick up for me.
I was making a mukbang.
What is that?
What?
Mukbang.
Yeah.
Somebody's gonna have to.
Boy, in these backgrounds, bro.
I can't believe I don't have my.
Mendoza, why your camera not on?
I don't know.
Mr. Mendoza, you have to turn on your camera to show that you're here.
Mendoza, you want to see me solve this Rubik's Cube in 5 seconds? Watch this.
Go ahed. We'll watch for a second because I'm trying to. Well, until
Watch this. Okay. I'm different. I'm built different.
Bro, you're not like that, bro.
Watch.
We're not that guy.
I'm that guy. Okay.
Okay.
We don't think so, but that's all right. Go ahead.
You want a round of applause? Cool. Awesome.
That was amazing, actually. Can you do it again?
So great. So
interesting.
I gotta scramble it up. I gotta scramble it.
Okay.
I'm still trying to find somewhere in here. This is unbelievable. I'm gonna have to start again. Thought it was here.
do anybody want to DoorDash me some food?
No.
No, thank you. No.
It's
Go
crazy.
eat you some noodles.
Stay starving.
I don't want no noodles.
Go drink some milk or something. I don't
You got
know.
posters. You can eat those.
What?
I see them. Eat those.
All right. Hold on a second here. Somewhere in here, I had a presentation and for some reason it got deleted. It figures today's been the day of technological glitches all day long. My internet went out right in the middle of class earlier today. It was horrible. Are you all here still? I was listening to you all talk and now.
Yeah, we're here. Well, I'm here.
We're just letting you speak, Mr. Mendoza.
Oh, are you?
Yeah.
You got that pig again. This time, oh, you got the Puerto Rican pig, is that right?
Yeah.
Do you have one for every single place on the planet?
You want to see?
Why do you have so many?
Frank, you're sweating, bro. I won't even lie. He's sweating.
He has a pig addiction.
He can't even eat pork. Oh my god.
Yeah, they're his friends.
That's what I said. Oh, he has one with all the countries.
Yeah.
The inclusive pigs.
Too bad you're gonna kill him and make him into bacon.
He doesn't.
He don't eat bacon. He don't eat pork.
I don't eat them either. I don't eat meat.
All right. It's only. I try not to eat pork, but I'll be honest. Every once in a while, some bacon. God forgive me. All right. Are we ready then? We're gonna have to start from scratch. I had it out, but I'm gonna have to. Oh,
We like the scratch. Remember, we like it. We like it like this.
yes, but I wanted to. There's a lot of detail. I didn't want to waste time. My brother is such, oh, he just keeps arguing. He said, oh, he doesn't stop arguing. Do you have,
Mr.
do you have
Mendoza.
brothers or sisters like that? They just want, they never stop with the argument.
Who doesn't have brothers and sisters like that?
I don't know.
Mr. Mendoza.
I only got one. That's him. Yeah.
I'm joined on my phone because my computer is not letting me join. But my camera won't turn on my phone.
All right. That's fine. Will, could you get your camera on? Connie, your camera. I hope you feel better, Will. I heard you were sick yesterday. And I bet you that's Sophia. Sophia, could you get your boyfriend off the screen and get your face on there or some part of your
Her boyfriend?
Manuel? Is that why you put his face on there?
Sir, no, that's
The better Manuel.
What is W?
Yes.
Wait, the better? No, I'm the better Manuel.
Dang.
oh, absolutely
First of
not.
all- first of
Absolutely
all,
not.
I'm
Well,
older. That's
If
number
you're
one.
the better one, where's your $1 million franchise?
Money is not all that's important.
Jamal, I did it.
I got to point out that all the Manuels have the forehead, though. You notice that? We all got the forehead, for the record. All right.
Oh,
Why is it? What's wrong with Sophia? Why did somebody read the text? What is she saying? What's WYM?
What you mean?
Oh, because you have it on your screen. You have Lin Manuel on your screen.
No, that's a
What? All right, let's go on and start and get some work done. Does Marshall have like giant eyes?
you doing office hours today?
Yeah. What's Marshall doing? How's he do that? That's so weird. All right. Anyways, yeah, I'll be here at 1 o'clock for office hours. I gotta say, yesterday's session went really well. I recorded. I was processing it online. I thought that this Explain Everything would have a copy of it here, but it doesn't. So I'm going to go ahead and start again. And Gabby and I and a young lady from first period got together and we discussed it. And I think it went pretty well, but we're going to go ahead and do it again. So you have a cell, as you know, you have a cytoplasm. Remember, this is the cytoplasm of the cell. And remember that there's an endoplasmic reticulum, which is a membrane that actually goes all the way around the nucleus. And remember that there's a rough endoplasmic reticulum and a smooth endoplasmic reticulum. It's continuous. It's a path for production of biomolecules. And you have a Golgi body, right? This is a Golgi body, an endoplasmic reticulum, the smooth and the rough endoplasmic reticulum. You have to remember that the nucleus has holes in it. So don't forget that. The nucleus is porous. It has these pores, nuclear pores, that allows stuff out and stuff in. It has this. So those are the basic components. That's what we're going to focus on with this particular discussion. We're going to focus on the nucleus. And we know that the nucleus, one of the big things it contains is the DNA. And of course, we know there's a lot of DNA in there, and the DNA molecules are very long. And in humans, there's 23 pairs, so 46 of these chromosomes. A chromosome is, and remembering that these are ribose sugars, connected sugar phosphate on the backbone from 5' to 3', and the other strand is the opposite of that, 3' to 5', because they're anti-parallel. We should know all that, right? So the first step in this process that we've been talking about making a protein is this idea of transcription. The second step happens in the cytoplasm. It either happens here in the endoplasmic reticulum, or it happens here in the cytoplasm. Either place, we call it it's the second step that we call translation. So those are the two big processes. So going back to the beginning here, let's take a look at transcription. Transcription is going to use the enzyme known as Rna polymerase. And that's an enzyme that makes RNA, a polymer of RNA. My dog, if you hear snoring, it's my dog in the background. So the RNA polymerase is going to read, it's going to read the DNA and make what?
RNA.
RNA. All right. That being said, you're going to look at the DNA. It's going to say, it's going to have, not all the DNA has genes. A lot of the DNA doesn't code for anything. Some of these genes are permanently turned off. There are some ancient genes in our DNA that go back to our earliest ancestors that are never going to be expressed again, theoretically. For instance, there are people that are born with tails. That's because they have certain genes that are turned on that never should have been turned on. They should have been turned off that weren't, and they grew tails. The rest of us, those genes are turned off normally. We could have webbed fingers, for instance. We can have, so there's all kinds of things that can happen. Oh, Frank got kicked out. He was the one guy that was going to let people in. So there are genes that have never turned on. They're what we call remnants of evolution. So in the DNA, we can find genes that we share with other ancestors that we know we don't express ourselves. We can find genes from ancestral viral attacks. We know that there's genes in our DNA that show evidence of ancient viruses. So the DNA holds a lot of stuff that we don't express, that is not going to be expressed. We also have genes that are not necessarily expressed at the moment, right? So there are some genes, like for instance, if you think about it, if you have a cell in your toe, you don't want that toe cell to express an eye, right? You don't want to grow an eye on your toe, do you? That would not be useful, correct?
Yeah.
That would not be useful. So we have to have a way to turn the genes on that we need on at the moment and turn other genes off. So the way we turn those genes on, we call that epigenetics, is you can turn them off by wrapping these long-term, they're off by wrapping them around histones, those proteins that we can wrap DNA, like curlers, like we mentioned in class, they're like wrapping hair around curlers. Those will never be turned on because the DNA that's in that particular section is wrapped around these big proteins called histones. There's a lot of DNA there. There's probably a lot of genes. Maybe this is a cell that's in your toe. It does not need an eye growing here. So the genes that require that are for an eye are wrapped around a histone and turned off. We don't have to worry about those ever getting turned on for the life of this cell, this toe cell, right? This is a toe cell. So that cell is turned off. That gene in the eye is turned off. We don't have to worry about it. So that's good. That's good news. But there are other genes that we have to turn on and off repeatedly, like for instance, insulin. If this is a pancreatic cell, let's take a look. Let's erase this toe and let's say make it a pancreas. If this is a pancreatic or a cell in the pancreas, then it has to be able to produce insulin. It has to be able to produce insulin when the body needs it. Otherwise, you're going to be diabetic. If you can't produce insulin when the body needs it, then you become diabetic. You can't control your glucose level. So we have to have a way to turn this on when there's high sugar in the blood. and turn off when there's low sugar in the blood. So, how do we do that? Well, we have this region before the gene that's called a promoter region. This is DNA, it's just DNA. That's called a promoter region. And it has specific properties that you could look at if you like. In AP, we'll look at them. But then you have another region of the DNA, and I'm just color coding them. That's all I'm doing. That we call the gene. This is the gene. So this is actually the coding sequence. This is the part that's expressed, the part that's going to be read and turned into RNA. That's called the gene. Then you have the termination sequence, the part of the DNA that says, okay, that's it, stop. So we call that the termination sequence. So there's a region of the DNA that stops it, terminates it. There's a region of the DNA that is the gene that's expressed. And there's a region of the DNA that's the promoter region that's, so you have like a book, like a sentence, you have the beginning, the actual message, and then the end, right? So in a sentence, the capital letter tells you that's the beginning of the sentence. The period tells you that's the end of the sentence. The sentence itself is the stuff in the middle. So that's how we're running. That's how your DNA reads a message. This is how you know that this is the gene you need to turn on. How do you signal that this gene needs to be turned on? Is you have these things called transcription factors. Transcription factors can be proteins, they can be ions, they can be RNA, they can be anything really. But the transcription factors, it depends on the species, and it depends on the species of living organism that you are, genus and species. And individual tissues might have different transcription factors. So transcription factors is signaled by the body, yo, I need this specific gene turned on. In this case, it's the gene for insulin, right? So turn on this. So these transcription factors are probably going to be proteins that are stimulated when there's high glucose, and you're going to turn on the gene for insulin. So what happens is the transcription factors bind, and that allows the RNA polymerase then, and I'm just going to, this is the RNA polymerase. The RNA polymerase is going to land, and it's going to start, it's going to land on the promoter region, and it's going to start to read, it's going to start to read the gene. It's going to start to read the gene. So now, if in the gene you have, let's go ahead and use a different color. If in the gene you have A, T, G, C, A, T, G, C, A, right? If that's your message, it's on the gene. Obviously, the gene is a lot longer. It could be thousands of nucleotides long. We're going to make it short because nobody has time for that, right? So if you start to read this, tell me what is going to be the RNA that's produced by this message? Somebody tell me.
UAC.
Hold on. UAC. Yeah.
G. U. A. C. G. U.
Good. Does anybody have trouble? Does anybody have trouble understanding how she did that? I don't hear anybody saying that they do. If you want to message me privately, you can direct message me, let me know. If you want to come during office hours and see how we do that, we can. Gabby came at 1 o'clock yesterday and we went over this stuff, and I think it was better for her. Gabby, did you find it useful?
Yeah.
All right, so it can be useful to meet with me if you're confused. So this is an RNA. Did I say mRNA?
No.
No.
No, I didn't. This is an RNA. I'm gonna go ahead and make an erase. I'm gonna erase some stuff here because I need more space. I hate doing it because I drew such a beautiful circle. Better than any of you could have done.
Are you doing shots, Mr. Mendoza?
Why did you have to throw
I
shit?
was wondering when I was gonna hear about that. I threw
Why
shade.
did you have to throw
I'm just
shit?
saying, like, why did you have to throw shade?
Oh, Ivy's here. Quiet.
80 Thursday.
All right. Yes, I was throwing shots. All right. So now in RNA, there's three options. Does anybody know what three kinds of RNA? This RNA, this is a gene, right? When, by the way, now transcription's done. That's it for transcription. That's all there is to it. Transcription is just reading the DNA and making an RNA. We're done. That's the transcript. This is the gene has now been expressed. We're done. We have expression. We have an RNA. Now, the question is, what kind of RNA? There's three different kinds, at least three different kinds. Actually, it's more, but let's just say three different kinds of RNA. What are the three?
mRNA, tRNA, rRNA.
RNA.
That's right. That's right. So we can make, so now we can, this could be an rRNA. This gene could be a tRNA. And or this could be turned into ultimately an mRNA. All right. Now, if it's an rRNA, it's just going to leave the cell through the nucleus, through the nuclear pores, right? Through these pores. Now, the rRNA is single, it's not double-stranded, all RNA is single-stranded, right? It's smaller than the DNA, so it's going to fit through the nuclear pores, no problem. Out it comes, no issues. That rRNA is going to go into the cytoplasm, and it's going to form these ribosomes. It's going to form these ribosomes. So what you'll get is one gene will make the proteins required for the large subunit, and one gene will make the proteins required for the second subunit, and the two together will make a ribosome. So a ribosome is RNA. It's made mostly of RNA, and it comes right out. That's it. There's nothing more to that. It's going to act as an enzyme, and that ribosome is going to make. In fact, I'm going to stop drawing it right there, and I'm going to erase it, and I'm going to move it over. It's going to take and move it over here because it'll give us more room. So this is a
You
ribosome.
said after transcription, it makes a histone.
No. Histones are used to turn genes off. They're used to wrap DNA around them. If the DNA is wrapped around a histone, it can't be read. Does that make sense? If you have the DNA,
Wait, can you repeat that?
if you have the DNA wrapped around this histone like hair around a curler, you can't open it up to read. See, this is open. This is called the transcription bubble, right? The DNA is not double helix here. It's opened up. The two strands are separated. That's the only way you're going to fit this RNA polymerase in there to read it. So the histone, the DNA that's wrapped around the histone is not going to be opened up. It's turned off. That's why you don't get an eyeball in your pancreas. You don't get an eyeball in your pancreas because the genes for an eyeball are turned off in the pancreas cell. Does that make sense?
Yeah.
Okay, so that's a histone. Now, what we're talking about here is once you've transcribed, transcription is just reading the DNA, not reading the DNA, and making an RNA. That's it. Your done. Transcription is done. Now, the only question is, which RNA did you make? If you made the rRNA, then all it does, the rRNA comes out here. It folds up into a shape. Some of it folds up into the large subunit. Some of it folds up into the small subunit. And the two of them together make a ribosome. That ribosome does translation. It's going to take the message from mRNA. It's going to make an amino acid sequence. All right. Oh, I can't believe it. I keep doing that. Give me a second. I have to move that over here. Let me group it. All right. So now, if it's a tRNA, then again, it's done. Your transcription is done. All you're going to do is it's going to come out here and it's going to form a shape that I personally think looks like a T. And that tRNA is going to carry an amino acid. And it could be any one of the 20 amino acids, but that's going to be defined by the anticodon that's on here, and therefore by the codon and then the anticodon. The anticodon just matches the codon. And that, let's say it's that. So that's the anticodon. Now comes, what if it's an mRNA? What if it's supposed to be an mRNA? Then what's going to happen is that RNA, there's going to be parts of it that are going to be cut out. So maybe this part gets cut out. Maybe this part gets cut out. And remember, it's much longer than this, right? So those parts get cut out. And then these leftover parts get put together. What I'm describing here is called, let me do it a different color so it's easier to see. Post-transcriptional modification. What that means is that it's post means what? What does post mean?
After.
After, what does transcriptional mean? After what? So after what?
After transcription?
After transcription. It's just that simple. What does modification mean?
Add.
Add, yes, or change.
Change.
Add, subtract, change. Modification means change. These three fancy words just mean changes after transcription. That's it. No big deal. So what are the changes after transcription? Well, we're going to cut out the things that are called introns. These are called introns. Going to cut those out, and we're going to splice the exons together. The green, the rest is called exons. So I'm going to go ahead and put it together. I'm going to add some more letters because I don't want to make a really super short one. So if I cut them out, what I'm going to have is U, A, U, G, U. I'm just going to go ahead and add some C, G, A. So I'm going to have that, right? And so what I'm going to do, then what's going to happen next is you're going to add a bunch of A's. The polymerase is going to add a bunch of A's to the end, just a bunch of them, a tail. It's going to have poly A tail. And it's going to add a 3' cap at the other end. That's it. Now you have an mRNA. Now this is the mature mRNA. Now, why does it change the code? If this is your DNA, if this is the message, why does your body change the message from the DNA? This is what you inherited from your parents. Why are you changing it to something that's different? Why does this post-transcriptional modification even happen?
So that it could leave the nucleus?
Ah, good. Yes. Part of it is because you can leave the nucleus, is correct. But also the reason that we change it, right? The reason we change it is so that one gene can give us more than one protein. If that allows us to be very complicated, much more complicated than, let's say, a prokaryote, a single. You don't need as many genes to get as much done. You can do more with less messages. It's like, have you ever compressed a file on the computer? Have you ever had a picture or a video that's too big to send and you had to zip it? Have you ever done that? Yes, no, maybe?
Yes.
Some people, yes.
Yes.
So you just compress the file. When you send it, you don't send the entire message. That allows you to send a lot more through the internet than if you didn't compress it. So by taking this message and allowing it to be interpreted or transcribed in different ways through post-transcriptional modification, you're able to get more options in the end than you would with just one single gene. That allows us to be much more complicated than the 10,000 or so genes that we have in our cells would allow us to be if we didn't do this. So this increases our ability to have these complicated things going on in our body that allow us to think about us as being alive, that allow us to have complex thoughts, complex memories, that allow us to even consider things like God and souls and etc. All that stuff is allowed because we have this unique ability to change the information in the gene in various ways. All right. So we have this mRNA. It comes out again, just like someone said, I don't remember who said it. It comes out here, and it's either going to have, now there's there's 2 points, 2 places it can go to get translated, right? Because now we're in the translation. Where are the 2 places? It's going to come here if it's going to leave the cell. So if this is insulin, the protein is going to be made in the rough endoplasmic reticulum. It's going to follow into the smooth. It's then going to go into a vesicle. It's going to go into the Golgi body, and it's going to leave the Golgi body and then leave the cell and go into the blood, because that's what insulin is supposed to do. It's supposed to go and signal all the rest of the cells in the body that you need to let sugar into the cells and out of the blood. So this is where it's going to be made if it's a protein that's leaving the cell, or that's going into the membrane, or that's going into another organelle. If the protein is going to be staying in the cytoplasm, then it's going to be made here. It's going to be made in the cytoplasmic ribosomes, the ribosomes that are in the cytoplasm, not the ones that are in the endoplasmic reticulum. But in either case, it's going to find a ribosome. So let's go ahead and take a look at that. Let's go ahead and make this over here. Let's say, what was the message again? It was, let me see, UAC, G, oh no, is that, no, I messed that up. UAU, do you see that? You see what I did? I went before, it was before the modifications. I got to go with the after. So UA, UG, UC, GA, A, okay? All right. So now it's at the ribosome. The mRNA is there at the ribosome. We need to translate it. How do we translate it? What do we need to translate it? Anybody know?
tRNA.
Yes, you're right. We need tRNA, and we need 2 charts. If we're going to do it in the classroom, we're going to need 2 charts. We need this chart here, right? And let me take this and move it over here and connect it all. We need that chart, and we also, or we could use another chart that you've used in class. I don't know which one the end of course people from the state of Ohio are going to give you. So we're going to look at both charts. All right. Now I'm going to take a break for a second. I got to go to the restroom real quick. I want you guys to go ahead and discuss amongst each other how you're going to take that mRNA and what amino acids are you going to make. Everybody try to make it on your own. I'll be right back.
That's
Everyone
so
try
small.
to
How are
make
we
it on
supposed
your
to see it?
I can't see.
So we not leaving in 1 minute?
1
Do
minute.
we
1
start?
minute.
So we not leaving in 1 minute?
Yeah, 1 minute
Yes.
remains.
Oh, it's 11:59.
Yo,
But
it's 12
o'clock.
It's
Oh, it's
12.
12. It's
I
12.
know.
All right,
Y'all
y'all
be
have a
easy.
good day.
I'm going back to sleep. Goodbye.
When the clock
Y'all
strikes
have a good
five.
day.
Anybody
You
going
guys
to
have a
office
good day
hours?
now. You guys
Bye.
have a terrific day, okay?
Yeah,
You
y'all.
too.
Yeah. Y'all be good.
You too.
Mr. Mendoza is gonna be shocked when he comes back.
Like, where did y'all go? No,
I
come
kind
back.
of want to stay here just to see that.
Exactly. But there ain't nothing he can do. They're already gone.
So guys, let's discuss this chart, shall we?
Yeah, it's art and it has letters
All
on
right.
it.
So
I can
start.
see it. I can see it a lot. I can see it very well.
Can you?
No,
No, you can't.
I know I can't.
But so it start.
All right. So for AUG, we make met.
Start.
Whatever that is, met.
I thought we made start.
Sorry, guys.
Oh, he's back.
Oh, thank
Hey,
Class
Mr.
is
Mendoza,
over.
how's it going?
All right, sorry about that.
Class is over.
Is class over? Oh, bummer.
Everybody
Actually,
left.
can you keep going because I
Yeah.
kind of still want
I'll finish
to.
I'll finish this and then I'll post it and then people can watch if they want. Also, I'll be here at 1 if you have any questions. So
Because
Yeah, I
I
might
gotta
pop
make a
in at
new
1.
page because I didn't know the circle had to be that big.
Man, I
I know,
literally
I-
erased my whole thing and I just started writing like word notes because I could not draw that.
Yeah,
I'll
I
post
didn't
it.
know the circle had to be that big. So
Yeah, I
gotta
couldn't
make
draw
a
any of
note.
that.
I'll post it right after, but let's take a look at it. We got you're right. I heard someone say AUG, and you're right. AUG is going to be the beginning. That's where we start. AUG makes what amino acid?
Met.
Met.
So MET is the first.
That's the only one I know by heart.
But we should read it. We should practice reading it, right? So let's take a look at both.
Wait, hold on, Mendoza. I could do the second one. Let me do the second one.
All right, but let's do this one first. AUG, AU, and then G is MET, and that's the start. See how you read that? Start from the center and go out. Now, in this one, oh, I don't know why it doesn't. Why didn't it?
You go left and then up and then you find it.
That's right. So when we go here, we go A, first letter's A, second letter is U, and the third letter is G. So obviously MET is red here, but it's A, U, and then G, and then so that's MET. It's also the start. That's the only, even though we have stuff before it, we're going to start at the first AUG. Even though there might be more AUGs as we go on, this is the only one that's the beginning. This is the start. Now, I say that you have to read the question carefully because some questions, as you see in the practice problems I gave you that are due on Monday, you'll see that the AUG, sometimes they have doing it, transferring this in the middle of the mRNA. So the second, what's the second codon? Second codon is UCG. The third codon is AUA, and then AAA, and then we're done, right?
Wait, so we don't touch the U in the beginning?
No, yeah, we don't. We read them. We start reading at AUG. That's what start means. Start reading.
Okay.
Now I say that, but if they tell you in the problem that they don't give you an AUG, they tell you it's in the middle of the problem, then you just start wherever it is. Does that make sense? But officially, when it works in the real world, AUG is the beginning. Anything before it doesn't count.
So you always start with AUG.
You do, but I said, but it depends on the problem. Sometimes they try. It's not really. They're not trying to trick you, but if they're asking you about a question in the middle of translation, not at the beginning, then they might not have an AUG in there.
Okay.
So don't get tricked. All right.
Okay.
So the first one's methionine. Now go ahead and find UCG. Do it on this one first.
Hold on. UCG.
Sir.
That's sir? Yeah, sir.
Yeah, sir, serine. Now what is it the same here? UCG.
UCG. Yeah, sir.
Yeah.
Still,
All right,
sir.
so the next one's, oh, I did it again.
Oh wait, I gotta write this. Hold on.
So the next one is SER, S-E-R, serine, serine.
So we do met.
Serine, right? Met, serine, they're together. They're now a dipeptide. Now the next one's what? A-U-A.
A-U-A. A-U. That's ill or I.
Yeah.
I don't know.
A is isoleucine, is correct. So leucine. The next one's AAA.
A. LS. LYS. List.
Right.
Lists, yeah.
Yeah, it's lysine.
Lice.
It would be lysine, but don't worry, LYS. You don't have to worry about remembering which one's which and all that. You don't have to do any of that. So, and you would keep doing that until you would reach what? What would be the stop? What would stop you?
We get to the end.
The
What
stop
is-
codon.
The stop
Termination
codon.
point.
Yeah,
They want the block boxes.
UAA, UAG,
that's right.
Um,
UAA, UAG,
U
or
UGA.
UGA, yeah.
Yeah.
any of those. So,
Oh, you talking about where the block is? Okay, I see.
so if I'm going to go ahead and change this, and I'm going to say, well, let's go ahead and make this UAA. Then that's what?
Stop.
It stops.
Over here.
But it's
let's-
also
What's UAA?
Stop.
So
Stop. There's no amino
so that
acid.
means you don't write nothing after that.
Right.
so-
anything after that. That's it. We're done. So this is the end. So when you reach a stop, it's either UAA, UAG, or UGA. That's it. You're done. You're stopped. That's a stop. I should have done the stop with it. Right.
and this is post-transcriptional modification, right?
No, this is what? What is this?
Is this translation? Is it translation?
Translation is correct.
Okay.
This is translation. What we're talking about here is turning an mRNA into an mRNA into an amino acid.
Oh, okay. Okay. Turning.
Sequence is
Oh, I literally have that. I don't know why I said
RNA into
turning a
sequence.
mRNA into.
Amino acid sequence. And you have a bunch of practice problems in your final review because you're going to have to do all this on the final next week. If you can
Okay.
do this, we're ready to go.
Okay.
Of course, you have to know all your cell organelles and all that. We'll talk about that tomorrow. All right,
Okay.
guys? If you have any issues,
Okay.
come and see me at 1:00.
Okay.
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 All
Bye,
right.
Mr. Mendoza.
Bye-bye.
Bye, Mr.
All
Mendoza.
right. See you, Mendoza.
Obasa, did you need me? Are you there, Obasa? I'm ending this. Not allow you to hear what I'm about to say to them because this is outrageous that I pay this much money and sorry about that, guys. That was very upsetting. My poor dogs are freaking. I don't know what happened. I reset all my routers and my router and my Wi-Fi, and it finally got to working again. All right. Well, give me a second. How much time do we even have left now?
We have like 10 to 9 minutes left.
Oh, good. Thank God. That's enough time, I think. It's going to be quick, but I'm going to post the rest of the video online. All right, guys? That really was very upsetting. That's my iPad trying to join. That's not going to happen again if I can help it. All right. I'm sharing screen. Am I sharing screen yet? No. All right, so the slower version is going to be online, all right, but I'm going to try to make this as succinct as possible. If you want more details, see me at 1 to 2. If you're confused, I'll be here from 1 to 2, or just watch the video and we'll try to do our best for tomorrow to review. The best option is for you to get that practice done with that test review for next week. All right, so we talked about large subunit. So we made the mRNA. If we're making rRNA, that goes into the cytoplasm. That never gets turned into a protein. tRNA never gets turned into a protein either. It also leaves the nucleus and comes out here to the cytoplasm, where it carries an amino acid to the ribosome. Okay. Now, when we're out here in the when the message comes out, it's going to start to read the message. Now we're in the second step. We're not in transcription anymore. We're in translation. We're going to read. Now we're reading in 3 codons at a time. Is that understood? We all understand that?
Yeah, I understand.
Everybody? Anyone else?
Yeah.
Okay.
Yeah.
So there's no, I just don't want any surprises with people not, telling me that later that they didn't get it or gee, I was confused on the test. So when you're taking the end of course exam, they could use either one of two charts. We're going to go ahead and use both charts to give you some practice. So the first one you see here, the first triplet code, where does it start? Where does translation start? What's the code, the codon that starts the process?
AUG.
AUG is correct. So the first AUG that ribosome reads is the beginning, AUG. Do you see that? I underlined it in green. So as soon as it sees the first sequence, AUG, that's the beginning. It could be a lot of stuff ahead of it. It doesn't matter. The first AUG, there could be other AUGs down the stream, but the first AUG is your beginning, your start. Now, I got to tell you that in some problems, not on my test, but on some problems in the course exam, I've seen where you start in the middle of the process. And I gave you some of those practice problems in your test review. But if you're asked to start from the beginning, it starts with AUG. So let's look at this chart and try to find AUG. Where do you think you're going to start? Can you see this chart?
Yeah, I can see it.
So everybody else can see it? Yes? No one has a problem? So where's the beginning? Where do we start? Where do we find AUG? Where do you think you're going to start? Did I freeze again? Or are you guys just quiet and thinking?
We're just quiet and thinking.
Where's the first? It doesn't tell you like the other chart. The other chart's easier to read. I hope they give you the other chart in the course exam, but this is actually not bad. And logically, it tells you should know. A is the beginning, so it's going to be the one that has the least number of options, right? So A. Then you go to what?
U.
Green U.
The green U is right. And then the what G?
Blue.
The blue G. So you go A, you go start from the center, move outwards. Start from the 5' and move to the 3' because because RNA is read from 5' to 3'. That's how you read it. So so your AUG is what is AUG? Methionine. And what is that? What does that signify? The triangle signifies what?
The start.
Start. Okay. So yeah, so that's it. Now let's read the easy. I think it's probably most kids like this one better, but I like both. I think they're both simple once you get used to using them. And of course, you'll have both on my test, and you could use whichever one you like. Okay. But on the end of course exam, I don't know which one they're going to give you. So I want to introduce you to both of them. So AUG, there's an A here. That's your first letter. Where's your second letter?
The U at the top.
The U at the top is correct because it says second letter. So somewhere in this column is going to be our AUG. The U is here. This first in this row is A. Somewhere here in this row is our AUG. In this row and this column. So somewhere in here is our AUG. And how do we find our last row? We look at the G in this box, in this row, in this A row. There's our third letter, the G, third letter, the G. And that's AUG. It's methionine. And it is our start. So that's our first amino acid is AUG, is methionine, right? So methionine would be our first amino acid it's going to add to the amino acid sequence. Is everybody okay with that? Did you see that?
Yeah.
Okay. So now you go to UAC. UAC. Let's go ahead and see if we can read it here. Who could read UAC and tell me what it is?
Isn't it TYR?
TYR, that's right, tyrosine. Very good. See how easy it is, easy to read, right? So UAC. You'll see that the fourth position is very wobbly. They call them wobbly. The third position of all the codons are very wobbly. So you get U is pretty much always going to be U. It's always going to be tyrosine is always going to have a U in the beginning of it. Do you see that? TYR always has a U. TYR always has an A. So Y, it has a U and an A. Tyrosine always has a U and an A. But then it could have a U and a C in the last position. Did you see that? So UAC is always UA, tyrosine is always UA, but then it's either U or C at the last position. So in any case, tyrosine is UAC. So all right. So we got methionine and tyrosine. Now what's the third? The third codon is what?
UAA.
UAA. So let's take a look at that one. Who can tell me what UAA is?
Stop.
The stop of it.
Stop. All right. This is a two amino acid protein. The first one's methionine and the second one's tyrosine. Unlikely that would be the case. And there it is, by the way, there's our two amino acid protein. So it's unlikely they would be that short. I just made it short just so that we could just for practice and how to read. The ones in the practice in your final practice in your review packet, you'll have longer amino acid sequences to translate or RNAs to translate. So let's go ahead and do those last two here as well so that you can see how to do it. We have UAC and UAA. UAC, I hope you see it's U is the first one here. A is the second letter, so somewhere in this block, and C, UAC is tyrosine, TYR. Then the next amino acid after UAC is UAA, and I go to U again, and I go to A, and somewhere in this block again, and the second A leads me to a stop. So that's how you do it. That's how you translate a protein, and that's what allows us to. Those are the two processes you need to know for the final. I'm sorry for the technological issue. I know it's time to go. Have a great day, guys.
You too. Thank you.
Yeah. Hey, how's it going? Carmen, I'm making you co-host, okay? Wait, why did Carmen even come in there? I think she thinks we're following the Thursday schedule. Heaven, I'm making you co-host.
Okay.
As soon as people come in, let them in. Myasia, you too. Could you guys turn your cameras on? Hey, guys, let's turn on our cameras. We letting them in, guys? I can't tell what's going on. Let's get our cameras on, ladies and gentlemen. Hey, Triton, do you play darts? Yeah, is that a dartboard on your wall, right?
That's my little brother's, yeah.
Oh, okay. I used to play darts when I was a kid.
Did you even hit the board?
Oh, it's so funny. Yes,
Thank you.
yes, I did. Yes, I did. Now, why is my camera not on? I turned. There you go. Oh, why is it taking so long for you guys to join in? Turn your cameras on. Walker, Colbert, Precious, Lee. Let's go on. Let's start this. All right. So I gotta tell you. Oh, what's this? Another last. All right. All right. So sorry. All right. So we have I think I'm recording, but let me record. Yeah, I am recording. Just so everybody knows you are being recorded. Someone put in the chat something. Yeah, go ahead and get your notebook, Precious, and then Walker, Graves, Lee, Colbert. Come on, turn your cameras on. I'm going to start without it, and if it's an issue, I'll call your home and we'll talk about it. Figure out why you can't get your camera on. All right, are you there? All right, just to make sure at least you there. Could you respond in a chat or speak or something? I know Precious went to go get her notebook. Christian, you there? Maya?
Yeah.
I'm here.
Okay. So let's go ahead and start then. I got to tell you one more time. I'm going to tell you every day, tomorrow as well, because I don't want any surprises. On Monday, your final review is due. It was an elective assignment over break, but you had this whole week to get it done by Monday. It was assigned to you, a week before we went on break. So you've had it for about a month now. It'll be a month and a week before when it's due. So bottom line, all that's due on Monday, and you can go to it. The assignment's posted in Schoology. Guys, it's going to help you get through that test. The second thing I want to say, I know I keep repeating myself, but I feel that if I don't repeat myself, people say, I didn't know. So let me be very clear. I am not accepting any late work after Monday. So if you have late work, get it turned in on Monday. All right. That being said, let's go on and go through this. So there's the whole point of DNA. We looked at DNA yesterday and we talked about it. And the whole point of DNA is that DNA has a message that helps the cell helps control the cell, helps the cell do what it needs to do. The way it does that is that the message, it first has to be transcribed. And that's so the first step to this process is from taking DNA, the message from in the nucleus, and making it do anything is to transcribe it. It has to be turned into RNA first. That's what transcribe means. It's turned into DNA. Make sure you know that. So what is it that allows DNA to turn into RNA? It's this enzyme called Rna Polymerase. You need to know that. It will be on the test. So DNA gets turned into RNA. But a lot of the DNA does not have a lot of the DNA it doesn't code for RNA. It has old genes that are left over from evolution. For instance, we have a gene that allows us to grow a tail. Most of those genes are turned off. We have it in our DNA. Some people are born with tails. Our fingers can be webbed like Aquaman, right? But those genes are generally turned off for us as well. So there's all these genes that we turn off normally, permanently. There's other genes that are turned off semi-permanently. There are genes, for instance, if you can imagine, the cells in your toes have the genes to make a brain. And we don't want to grow a brain in our toes, do we? What do you think, Christian Walker or Christian Graves? What do you guys think?
No.
Grow a brain in our toes? No, we don't. So we turn those genes off semi-permanently. And the way we do it is we wrap them around a histone and we leave them put away. We don't ever unwrap them. There's other genes that we need to turn on sometimes and turn off at other times. For instance, insulin. You want insulin in your blood if you ate a candy bar, but you don't want insulin in your blood if you haven't eaten for a while, because if you do, you could get low blood sugar and pass out and go into a coma. So it's important that we turn that gene off when we don't need it. So how does a cell do that? It uses the rest of the DNA that's not a gene. The green part that you see here is a gene. The stuff before it in yellow is what we call a promoter region. So if the cell needs insulin, for instance, it'll send out transcription factors that tell the cell, turn that gene on and make insulin. If it does not need them, then it won't send out the transcription factors and that gene is not turned on. So that's how your cell regulates expression of a gene. A gene is a sequence that leads to an RNA. Expression means making an RNA molecule from that DNA. If there's no RNA molecule made from the DNA, then that gene is not being expressed at that moment. All right. So you have the signal, the transcription factors land on the promoter region. The promoter region binds with the RNA polymerase. There's the RNA polymerase on the promoter region, and it starts to read the DNA. When it gets to the end of the message, that's the beginning of the message. That's the promoter region. So the promoter region acts like the capital letter in a paragraph, or acts like the indentation in a paragraph, or acts like the page numbers in a book. It's what tells you this, or tells yourself this is the beginning. That's what the promoter region does. Is that this is where you start to read here. That's what's called the promoter region. At the end, you have a termination sequence. That's the period at the end of the sentence. That's the end. That's the end of the story, the end of the book that says the end. So the termination sequence is the end of the message. The beginning of the message is the promoter region. The end of the message is this termination sequence. That's all in your DNA. So your gene is very well defined. There's a promoter area and there's a termination sequence. So it tells you beginning and end. Now the RNA polymerase is going to read that message and it's going to turn it into DNA. So everywhere it sees an A, it's going to put a what in the RNA. Where it sees an A, it's going to put a what in the RNA. Somebody answer.
U.
Correct. Everywhere it sees a T, it's going to put an A. Anywhere it sees a G, it's going to put a C. Anywhere it sees a C, it's going to put a G. So that's your RNA. So not all RNA gets turned into proteins. Some RNA, ribosomal RNA, is turned into just that, RNA. It doesn't go any further. That rRNA, that ribosomal, if this gene were an rRNA, that RNA, after it's made, you see here it's made, A goes to U, T to A, G to C, etc. So there it is, there's the RNA. If this is rRNA, it's going to leave the nucleus through these nuclear pores. Your nucleus is like, the nucleuses of your cells are like Swiss cheese. They have all these holes in them. We call them nuclear pores. And these nuclear pores allow RNA to leave the nucleus. DNA can't get out because it's really big and it's double-stranded. So it can't fit through the holes, the nuclear pores. But RNA can. It does leave the cell. So our RNA leaves the cell. There's two genes, one for the large subunit, one for the small subunit. And they come together to form a ribosome. And that ribosome is the enzyme that makes amino acids later in the process. So our RNA has its own gene. The large subunit and the small subunit have their own gene in the nucleus, in your DNA. Now, another type of RNA, if this gene codes for a tRNA, there's at least 20 different tRNAs, and we know there's more. There's something like 60 different tRNAs, but let's go ahead and think of at least 20 tRNAs. These different tRNAs, if this gene were a tRNA, it would just leave the nucleus. It would come out here, would fold, it would bend on itself, and it would find an amino acid to take to the ribosome. That would match the anticodon on the other end of the tRNA would match the codon in the ribosome, and the first amino acid would be brought to the ribosome. And we'll look at that in a minute. We've looked at it before in class, but that's how tRNA works. Now, if this RNA, because there's, as I said, there's three possibilities once your gene gets turned into an RNA. It could either be an rRNA, a tRNA, or it can be an mRNA. The mRNA has to have the message from the gene. But the message of the gene does not go into the mRNA without changes. So your cell changes the message. We call these changes post-transcriptional modification, after transcription. So let's be clear. Once you go from a DNA to an RNA, transcription is over. You're done. You've transcribed the gene. The question is, where do we go from there? So we can take the RNA, and that could leave the cell, and our RNA could leave the cell and be a ribosome. tRNA could leave the cell and carry the amino acid. But if it's going to be an mRNA, it has to be changed. And these are the changes that are going to happen. It's going to take some of the sequence and just cut it out. We call that cutting out the introns. The introns are cut out. The exons are then spliced together. You put a 3' cap on it, you put a poly A tail, and now we call that an mRNA. That's it. No big deal. Now the mRNA leaves the cell. We're done with transcription at that point and post-transcriptional modifications at that point, and everything else happens in the cytoplasm or on the RER, rough endoplasmic reticulum. At this point, there's another decision-making point. This is another decision-making point. If out here in the cytoplasm, if the RNA, if the mRNA has the message is going to be for a protein that's leaving the cell, testosterone, estrogen, insulin, all have to leave the cell and go into your blood out here. So they have to be made in the rough endoplasmic reticulum because they have to go into the rough endoplasmic reticulum, then into this, into a vesicle, into the smooth endoplasmic reticulum, then into a vesicle, then into the Golgi body. Then in the Golgi body, it gets changed. It gets sent to wherever it needs to go. Whether it's going to be a membrane protein, it's going to be embedded in the membrane like a channel protein or a signal protein or anything like that. Or it's going to be a protein that's going to be put into the blood, or it's going to be a protein that's going to be put into an organelle. Whatever the case, any of those proteins are going to be made in the rough endoplasmic reticulum. If the protein is going to be used in the cytoplasm, then that protein, then that RNA, mRNA is going to meet up with the ribosome, and it's going to make the amino acid right here in the cytoplasm. In both cases, the process is the same. So let's go ahead and look at the process in detail. So the ribosome reads the message, and what's the beginning? What's the first? Wherever the beginning of the message is always AUG. You should know that. You should be don't be surprised if you get tested on that. AUG is the beginning of the message. Now, I'm going to give you both of these charts on your test, but I'm not sure which of these charts the state of Ohio is going to give you. One, I think, is pretty easy. That's this one. If they give you this chart or one like it, I think they're both easy, to be honest, but you have to get practice using them. So that's why I suggest that you do your practice that I've given you for your final. Of course, it's due on Monday, but it's really going to be important you do that practice. So AUG, can anybody read AUG off this chart and tell me what AUG is? What is AUG? When the ribosome, say, go ahead. Met, correct. And so I'm assuming you did it by looking at the first letter A here, that's somewhere in this, the message is somewhere in this column. This row. U is here in this column somewhere. So that means that AUG is somewhere in this box. And then G is here at this last row in this box, which is AUG, met. All right. Let's look at this chart and see where can you find AUG here?
Is it thou?
No. You need to get, probably haven't had practice at this. You shouldn't be, it's no shame in getting it wrong because it's something that's new. So please don't worry about being wrong. Go ahead and try it.
Are we saying what the thing is, or?
Yeah, what is AUG?
Met?
Met.
Yeah, it's going to be the same. It's going to be met, but how did you read it? Hopefully you started here at the A, then you went to the U here, and then you went to the G. So AUG, starting in from the 5 prime, moving towards the 3 prime, AUG gives you methionine. What is methionine? What does this triangle mean? Look at the triangle. What does it mean? What is methionine?
Start. Where to start at?
That's right. That's the start. That's correct. So methionine is start. So the first amino acid in the sequence is methionine. Now, what's the next amino acid sequence? Or the next codon, not amino acid sequence, but the next amino acid. What's the next codon?
UAC.
UAC. So let's go try to find it here. Can you find UAC here? If you can't find it, you need to try to see me during office hours, or you need to get practice.
Is it tier? The T-wire?
That's right. It is T-wire. Good. So it's UAC. UAC is TYR. It's pretty straightforward, right? Now we can go down here and find UAC here.
CYR.
That's right.
Yeah, I like this one a lot better.
I like this one too because it's colorful, but UAC goes across this way, and that's tyrosine as well. So they both work. Whatever one you prefer, I'll have both on the test for you. So that's so far we got what? We have methionine and we have TYR, right? Tyrosine. Correct? Correct? Yes? No?
Yeah.
So methionine and then tyrosine. What's the next one?
UAA.
UAA is correct. So we come here, who can read what UAA is?
Stop.
Stop.
Oh, yeah, so stop. So we're done. There's no amino acid connected to it because it's done. What about this one, UAA? Can you see it? Can you find it?
Stop.
Same
Stop.
thing.
UAA. It is stop. And there it is stop. Yeah, I'm glad that you know that you see it's the same thing. My thing is, can you read it? Can you look at this chart and read it? Because again, on my test, you'll have both of them, but on the end of course exam, I don't know which one they're going to give you. So you're right there. This message produces a dipeptide, two amino acids, a methionine, then tyrosine, and you're done. So that's it. That's transcription and translation. Do you all agree that it's pretty straightforward? It's not that difficult?
Yes.
All right. So you need to get that practice done so that you don't make a mistake on your test, because getting any of this wrong on a test is a wasted opportunity. How much time do we have left in class? Does anybody know? I can't see my computer, so at 10 minutes, is that what you're saying? Yep.
Yeah,
Okay.
like 10 minutes. It's 9:28.
All right, excellent. So I had an idea of going through some osmosis diffusion questions. Is that where you want to go next in this review? Because we only have half an hour each day, and we only have one more day after this. I want to give you whatever you think you need the most help with. I'd like to help you with it. That was cute. Osmosis diffusion, do you guys want to do that?
Sure.
Yeah.
Okay. Please do that review, or you're going to find, here's my worry. My concern is you're going to start to do the review on Saturday, and you're going to run into all these problems. You're going to start texting me, messaging me. I'm not going to answer because I'm going to be busy. And then you come Monday, you're going to be completely confused, and the test is that week, right? I don't know when it's going to be because it depends on what our schedule is next week, whether we're in school or still virtual. So what the heck, man? You got to get those practice problems at least looked at so that you can try to deal with it. All right. So let's take a look at some osmosis diffusion questions. So you have a cell, and sometimes they'll give you an animal cell, and and sometimes we'll look at 3 or 4 different kinds of cells. They can give you a plant cell, right? Or they could give you a baggie. They can give you a little baggie. Or they could give you a YouTube even. I've seen the YouTube on these standardized tests. And what they'll do is in the YouTube, they'll put a membrane. It's a semi-permeable membrane. And they'll start you off with water on both sides even, right? So they're both even. And I have to give myself some more space here, so let me move this over. Then the animal cell, what they'll do is they'll give you a beaker, and they'll put water in it, and then they'll put the cell in the water. And the plant cell that I've seen in any case, I've actually just, for the most part, they just kind of give you the plant cell. Again, it's a plant cell, so it has a. All right, so those are kind of the three that they give you. And they'll put them on 3 different conditions in general. And they'll ask a question if, and they'll obviously with the YouTube, they'll put in side A and side B, side A and side B, side A and side B. On this, it's either going to be outside or inside, right? And they'll ask you if A is 20% salt and B is 50% salt, and the membrane is not permeable to salt, what's going to happen? Can someone answer that as far as the YouTube is concerned? Could someone answer that?
Answer what?
I could say, which one are we answering?
All right. So again, if A is 20% salt, so this is 20% salt on this side, and B is 50% salt on this side, and this membrane doesn't let the salt go through, will not let the salt go through, what is going to happen?
Is it the what's that word? Does it start with the I? I think.
Starts with an O.
Osmosis.
Osmosis, correct. So osmosis is going to happen. In which direction is water going to flow?
The slide with the higher concentration solute.
That's correct. Excellent. And that's how you remember it. It goes toward what if the membrane doesn't allow the solute through, and the solutes are different concentration, then the water is going to move towards the area of higher concentration of solute. The other way to think of it is that if this is higher solute, then it's lower water, right? That's another way of thinking about it. But in any case, that's correct. The solute, it's going to move, water's going to move in this direction. But they may not ask you which direction of water is going to have. They might ask you, they might have A, the water in B goes up, C, the water in A, the water in the A tube goes up, B, the water in the B tube goes up, C, they no change. What do you think is going to happen, A, B, or C?
A.
Which water level is going to go up? The tube, the water in A tube, the water in B tube? You think the water in A tube is going to go up? Why do you think that?
Because that's a lower salt concentration.
Yeah,
Like I'm looking.
But we said water, look at the arrow I drew. We said the water is going to move towards the area of higher salt concentration. So it's moving over in this.
Oh, I was thinking the other way around. Okay.
Where's the water going to go up? It's going to move towards the area of higher salt concentration. So the water is going to go up the B tube. Is that understood to everybody? Everybody see that?
Yeah.
Okay. Let's move over to the plant cell for a second. What if the outside here were 20% and the inside were 50%? What's going to happen to that cell? If the
Can
water,
you repeat the question?
if the water outside is 20% salt, again, the salt does not pass through the membrane, and inside is 50% salt, what's going to happen?
Water is going to go inside the cell.
That's right. Water's going to go inside the cell. What's one? So it's going to what? Swell. So the answer, water will go inside. Possibly burst the cell.
That one's called hypertonic, right?
Inside is hypertonic. This side's hypertonic. The salt outside is hypotonic. So the cell was put in a hypotonic solution. So the cell was put in a hypotonic solution. Now, what if this inside of this cell is 50%, but the water outside is 20%? This is a plant cell now. It has a cell wall as well as a cell membrane. What's going to happen to this? Where's the water going to go?
Wouldn't the plant like droop, like fall to the side because there's too much water? That would go inside the cell or something?
It's a good guess, Precious. That was a good guess. You do remember the drooping and the stiffening, right, of plants. So that's good. It's a good memory. But the question is, let's focus on the water. 50% salt. Here,
It's 39, Mr. Mendoza.
All right, try to finish this first. And 20% salt outside. In which direction is the water going to go?
Inside the cell again.
Inside again. And that's going to increase Tuger pressure. It's not, it'll swell. It'll swell the cell. That's true. And the cell will push up against the cell wall. That will stiffen the plant, right? That will make the plant stand straight up rather than droop, because the outside of the cell has more water and less salt. All right, so I'm hearing it's 39. When are we leaving? When are we out of here?
No.
Yeah, right now.
Oh, right now. All right, guys. All right. I hope this helped. We'll finish this osmosis discussion tomorrow. Please review it so you have better. So it's a better experience for you tomorrow. Good luck.
Have a good day, Mr. Mendoza.
You too. Bye-bye. Lee, did you need me? Jayla? Terrell, did you need me? Are you there, Jayla? Are you muted?
No, I don't need you. I'm just switching over.
All right. Have a good day.
Too.